Great. Well, thank you for the opportunity for me to speak with you all today. I became intrigued by the microbiome and its contributions to our health ever since residency. And when I did my fellowship at OHSU, I found it really interesting that just like DVT prophylaxis is part of every patient's order set, at OHSU, every single patient gets a dose of Nancy's kefir as a medication twice a day as prophylaxis. And I thought that maybe they were onto something. And it made me get really interested in the microbiome and how it influences our health and vice versa. So as we know, there's about 100 trillion microbes that are on or in our bodies, and those include over 1,000 different species. And these species encode more than 500 times the number of genes than we have in our own genome. And so you can really know that there must be interactions between our health and the microbiome. The microbiome has been studied in other diseases as well besides obesity, and wanted to share a little bit about it just to get you intrigued. This was a study where they took esophageal mucosal swabs from patients who were healthy, who had Barrett's esophagus, or who had esophageal adenocarcinoma, and investigators were able to see that they had markedly different microbiome profiles depending on if they had cancer or not. Sorry. Yeah, I won't. And when they looked at patients with inflammatory bowel disease, they also found that they had very different microbiome profiles compared to those without disease. This was similarly seen in babies with necrotizing enterocolitis, and those with NEC had different profiles of their gut flora compared to healthy babies. And even in mice models of burns, there's a very marked difference in their microbiome profiles. Maybe more interesting than just what their profiles are is just what interventions can be done to show differences. And it's really interesting that in randomized control trials, by giving a probiotic or as well as a prebiotic to infants in the NICU, they were able to decrease rates of necrotizing enterocolitis, NICU stays, sepsis, and even mortality just by giving them a little, yogurt and probiotics. Similarly, when you had intubated patients in the ICU, giving them a probiotic versus a placebo decreases the incidence of ventilator-associated pneumonia and led to shorter hospital and ICU stays. And even more broadly, giving probiotics to all patients decreased the rate of C. diff-associated diarrhea with a rate of 1.5% versus 4%, or a total of 60% decreased risk. And even after colorectal surgery, giving probiotics can even decrease superficial and organ space SSIs and decrease other types of infectious etiologies. So given this data, it's really, quite remarkable what the microbiome can do and how we can influence it. And so today I wanted to briefly talk about some topics in terms of how obesity and the microbiome interact, how these microbiome changes might be changed after bariatric surgery, and start to talk about maybe some roles for interventions for obesity. So as you all know, obesity is really on the rise. More than 40% of all Americans are obese with a BMI over 30, and nearly 9% of the population has a BMI over 40 or qualifies as severe obesity. So it really affects our entire patient population. Obesity is associated with many diseases and can lead to premature death, increased cancer, and really results in worse surgical outcomes for all of us, not just bariatric surgeons. And so optimizing the care for these patients is really in our best interest as providers. So to delve right in, there's been multiple studies that show that there are many factors that influence our microbiome, such as where we live, our own genetics, and how much we exercise. But even from birth, whether you're delivered by cesarean or vaginal delivery, it really influences our flora. Similarly, medications such as PPI use, which changes the acidity of our stomach and therefore can cause bacteria not to be killed by gastric acid can influence the microbiome as well as our diet. And studies, for example, have shown that people who eat lots of meat actually have a change in their flora so that the bacteria are better able to break down those animal proteins. Of all the microbes in our gut, they're generally divided into 6 phyla, and of which these phyla, the Firmicutes and Bacteroidetes make up the vast majority of our gut microbiome. And the Firmicutes group's common bacteria are things like Clostridiols, Lactobacillus, Faecalibacterium, and the Bacteroides are things like Bacteroides. So to look into the earliest animal studies, they found that when they had germ-free mice compared with those conventionally raised with bacteria, germ-free mice actually weigh less and have much less fat compared to those with a microbiome. Additionally, when they took the microbiome from conventionally raised mice and transplanted them into germ-free mice, those mice gained a lot of weight and increased their body fat as well, suggesting that the microbiome directly affects our body composition. And this was despite the germ-free mice eating more food than those with bacteria. Next, researchers studied wild-type mice compared to those with genetically induced obesity with the mutation in the leptin gene. At baseline, those with the mouse model with obesity are generally larger, as the model suggests. And when they took the microbiome from these 2 types of mice and transplanted them into the germ-free mice, those that received the obese microbiome gained significantly more weight and had more body fat, telling us that the microbiome can directly influence their body size. And furthermore, they wanted to see, well, how does that actually translate? How does the microbiome potentially do this? And for their studies, they took the microbiome from these mice and profiled them of all the different bacterial phyla, and found that those that were obese had a much higher percentage of Firmicutes compared to the lean counterparts, and that they had a decrease in their Bacteroides bacteria. And that this ratio, or this Firmicutes to Bacteroides ratio, increased. So they found that this was true across not only genetically modified strains, but they used a model of diet-induced obesity, where they fed mice either a standard low-fat, high-carb diet versus a Western high-fat, high-carb diet. And this led to increased body fat in the mice, but this also translated to this increase in Firmicutes and decrease in Bacteroides, very similar to other mouse models. And furthermore, they looked at the diversity of the bacteria within the stool, and found that those mice that were more obese also had decreased diversity in their microbiome. So while these differences in bacterial distributions are seen, one of the biggest questions is, well, how do those microbial differences mechanistically increase body fat or body mass? And one of the proposed mechanisms is via the production of short-chain fatty acids by the colonic bacteria. And so all of us, when we eat fiber, the undigested fiber gets fermented by the bacteria in our colon, and they produce 3 main short-chain fatty acids: acetate, propionate, and butyrate. And these fatty acids, all the residents know that butyrate gets used by the colonocytes for energy, but they really have much broader roles in being used for cholesterol synthesis, to be used for gluconeogenesis, lipogenesis, protein synthesis. And they also have a lot of effects on signaling as well, in terms of influencing inflammation, influencing the intestinal barrier, affecting motility, and even immune regulation. And so it's proposed that the alteration in these short-chain fatty acids may be contributing to the differences we see in body mass. And so the researchers took their experiments in mice one step further, and they took the stool from lean versus the obese mice and found that the obese mice actually had greater production of short-chain fatty acids, including acetate and butyrate. And when they did calorimetry on the leftover stool from the mice, they found that the obese mice actually had less calories in their stool, telling us that they were able to extract more energy from the food that they ate, and that this change is really seen in their microbiome. And furthermore, they looked at the genes expressed by the bacteria in the animals and found an upregulation of the microbial genes that are involved in carbohydrate metabolism and sensing, telling us that there's broad effects in how the microbiomes are working. So although those animal studies are interesting, we want to know whether these changes are seen in humans as well. And one of the earlier studies looked at 12 obese people versus 2 lean, and found that patients with obesity really did have higher Firmicutes and lower Bacteroides percentages compared to the lean controls. And when they put these patients on a diet, it actually translated into an improvement in their microbiome profile more to the lean comparison. And when they also looked at the percentage of body weight over time compared to their bacterial profiles, they found that there's a direct correlation with how much weight that they lost and their microbiome profiles. Similarly, another larger study looked at fecal samples from 61 people of varying BMIs, and when they did microbiome profiling on these 61 people, they found that with increasing BMIs, they saw a rise in the Firmicutes proportions and a drop in the Bacteroides, very similar to these animal studies, and to sort of reinforce what was seen in the animal models. And so there's an overarching theme based on these studies that the imbalance or balance of Firmicutes and Bacteroides can really be seen in obesity, where the Firmicutes goes up and the Bacteroides goes down. So while many studies have shown these findings, there are obviously some contradictory data as well. And one study looked at fecal samples from 97 people and found essentially the opposite findings, and that as BMI increased, they found the opposite, that Firmicutes decreased and Bacteroides went up. Another large study from the Human Microbiome Project found that there was no association between the Firmicutes to Bacteroides ratio and BMI, although this study did not include anybody with higher classifications of obesity. Similarly, another study of 40 patients really saw no association between this bacterial ratio and BMI, really telling us that, people really don't know whether whether there's a simple taxonomic signature of obesity and whether this really exists, but that there also might be other confounders that might explain these differences, in that patients with diabetes or metabolic syndrome have certain profiles that are contributing to their differences. How much does diet weigh in? How much does the fasting influence their microbiome, and did these studies account for when stool samples were taken? But also, we know that the microbiome effects on obesity might be mediated through other functions other than just their composition, such as their ability to influence metabolism and also the metabolites that the bacteria produce. So instead of looking at just those 6 phyla of bacteria, other researchers have really looked at the microbiome and obesity in different ways. One study actually found that when they looked at the profiles in 39 patients from 6 different countries, they really found a different way of describing the microbiome and found that people have different enterotypes, meaning that that people fell into different groups based on who had the highest abundance of these different types of bacteria. And these different types of bacteria which dominate have a different ability to harvest energy, and they found that this was even more influenced by the diet intake more than the BMI. So in addition to that, other researchers have also looked at microbial diversity. And so some papers studied 61 women who had obesity who were getting worked up to undergo bariatric surgery, and they found that a direct relationship between the body mass index and the microbial gene richness. So as the BMI increased, patients had a decreasing bacterial diversity, and that this correlated with fat mass and the trunk fat mass ratio as well, telling us that not only composition might be affected, but the diversity and the mixture of our bacteria. So there's been multiple studies that have looked at what interventions could influence these findings, and some small studies, just to sort of present to you, include a double-blinded randomized control where they took 75 patients who either were receiving a very low-calorie diet over 8 weeks, and they gave them probiotic supplementation or not. And they found that with the addition of probiotics, patients had a significantly lost more weight than those without the probiotics. And they even found decreased inflammatory markers like CRP and TNF-alpha and decreases in leptin. Other studies have also shown that when Lactobacillus graceri was given over placebo, they also had greater BMI change compared to controls and decreased body fat on CT imaging. And another sort of potential addition is something called a prebiotic, which are indigestible carbohydrates like inulin that are essentially fuel for our bacteria that can feed our gut bacteria. And the addition of probiotics and prebiotics together might have added benefit. And this study showed that when this prebiotic was given to diabetic women, they found that it could significantly alter their metabolism and their diabetes. So when taken all together, these studies really tell us that while there are studies that suggest that there is an imbalance in the types of bacteria in our gut, especially in obesity, that the story is much more complex. It's not just that the composition is different, but there's something called a microbial dysbiosis, where there's not only an imbalance in the flora, but there might be changes in the local distribution of the bacteria, how much is in the colon, how much is in the small intestine, and that there might be a functional a difference in the composition and the metabolic activity of our microbiome. And so the question is, well, does bariatric surgery reset that imbalance, and does it alter the microbial diversity and dysbiosis to make it more beneficial? So just to sort of review for everyone, the two most common bariatric surgeries are the sleeve gastrectomy and gastric bypass. We don't perform gastric banding anymore, but some of the studies I'll talk about do include this. We know that bariatric surgery results in a significant weight loss for our patients over the long term. Patients do regain a little bit of weight after they reach their lowest weight, but overall there's a good sustainment of their weight loss. So one of the studies looked at 10 patients who had adjustable gastric banding and 14 who underwent gastric bypass, and they looked at the fecal microbiome of patients pre-op and at 1, 3, and 12 months post-op. And they found that at baseline, all patients had a decreased bacterial diversity based on gene richness. And with both surgeries, both the adjustable gastric banding and the bypass, they had marked increases in their microbial richness. And interestingly, those that got bypass had even lower baseline richness before, but that the rise between the two were not significantly different. So it's really affecting their microbiome. Surgery is somehow. Another study took a look at 5 patients who underwent sleeve gastrectomy and looked at their stool pre-op and at 3 and 6 months post-op. And more similar to the animal studies, they did see that after bariatric surgery, after the sleeve, they did see significant decreases in the Firmicutes phylum and increases in the Bacteroides. And so that tells us that maybe we are recapitulating some of the things that have been seen in mice. They also found that when they looked at the actual expression, gene expression in the fecal samples, that there were elevations in the pathways involved in butyrate or short-chain fatty acid pathways, although there was no significant differences in the actual measured amounts of fatty acids in the stool. One of the other more recent studies also looked at patients who underwent bypass, only of 6 patients. They found a significant decrease in their BMI over the 3 months. But when they looked at the microbiome pre and post-op, there were significant changes in all of the bacteria. Namely, the Firmicutes decreased, Bacteroides was actually had no particular change, but that the ratio did decrease similar to the animal models. But even more confusing or complicating to the picture, that there were significant differences in the smaller proportions of the bacteria, the proteobacteria and the verrucomicrobia. And so it's not just the 2 main phyla that are affected, but it really affects all different types of bacteria. And other studies have also looked at other patient groups. One had 12 patients involved. They saw that patients dropped their BMI by 6 months, and that their diversity increased over the course of their 12-month study. And when they plotted the types of bacteria present at baseline in blue over time, so 3 months in orange and 1 year at green, they saw a significant shift in their microbiome profiles, and that they also found changes in their proteobacteria and fusobacteria as well, telling us that it's just a very complicated picture. Similarly, another study looked at 7 patients who underwent bypass compared to obese controls matched by pre-op BMI, and they found they looked at actual specific bacteria, not just the phylum, and found that there were 29 different bacteria that had significant differences after surgery, of which 3 of them were Firmicutes species, and there were also increases in proteobacteria. And when they looked at the gene expression in the fecal microbiome of these patients, they found lots of gene enrichment for genes that are involved in nitrogen and fatty acid metabolism, telling us that it's really affecting not only the types of bacteria, but the functional ability of the bacteria as well. Furthermore, they took stool from these bypass patients and transplanted them into a germ-free mouse model, and they found that those given the bypass patients' fecal microbiome, actually that the mice gained less fat compared to those who obtained the stool from obese controls. They had higher lean body mass, and this is despite no differences in food intake. Furthermore, they found that the mice that received the gastric bypass microbiome also had decreased respiratory quotients, telling us that somehow the microbiome is resulting in a change in how they're utilizing their carbohydrate and lipids as fuel. And just to complicate the picture even more, to make this confusing for all of us, the studies have also looked at patients who have regained weight after gastric bypass. So we all know that patients can regain some degree of weight, but that this study of 8 controls versus 12 who underwent bypass without weight regain and 14 that did regain weight, they found that patients over time maintained their microbiome diversity over time, so that this was still significantly different. But in blue is essentially patients who regained weight, and that they have very different microbiome profiles depending on whether they regain weight or whether they maintain their weight loss. And when the researchers looked at specific bacteria, they found that there were different profiles of specific bacteria as well based on their weight profiles. So the question is, even though we see differences in profiles, is there something deeper to explain these differences? And really, what comes into play is the functional ability of the microbiome, and that includes metabolomics and the metabolites that the microbiome is producing. And so this was one of the first studies to really describe the metabolomic and effects of the microbiome after bariatric surgery. And initially they found that patients who underwent gastric bypass had different profiles and had increases in 4 different groups of bacteria. But they took the stool samples from these patients and they looked at the metabolites in the actual stool samples and found significant associations between different metabolites in the stool for patients who underwent gastric bypass versus those that didn't, really telling us that the functional output and the metabolism produced by the microbiome is very different. And other studies have actually looked at patients who underwent sleeve versus controls and found that there's a complete change in the gene expression of these bacteria in the gut that are involved in multiple metabolic pathways that might explain differences in weight loss and differences in obesity. And so when we take that all together, I find this quite confusing, and I can't keep the bacteria straight. But I think what we're trying to understand is that it's not just the bacteria, but it's their metabolites and what they're doing for signaling. And when we really kind of delve into it, the microbiome has different effects. We already talked about short-chain fatty acids and how the bacteria in the gut have a differential production in short-chain fatty acids, but microbes really have many broader abilities in the body. Namely, they take our bile acids and affect our bile acid metabolism, and these bile acid metabolites affect direct signaling in our gut and in our liver to affect things like diabetes and glucose management. And the microbiomes also take things like carnitine and choline from the animal proteins we eat and create metabolites like trimethylamine and oxide, or TMAO. And these metabolites can influence things like our cardiovascular health as well. And really we're only really beginning to understand all of the metabolites created by our microbiome. And this is sort of the goal of the next step of microbiome research going forward. And so there's really a lot to be learned in this area. Well, besides learning about what exists or not, the next question is, well, how can we influence this, and how can we sort of potentially change outcomes in patients with obesity? And there really haven't been that many interventional studies done. One very small study included 30 patients who underwent gastric bypass, and they randomized them to obtain a prebiotic combined with yogurt or yogurt alone. And they found that there were no significant differences in BMI or weight loss after gastric bypass. Another study of 9 patients only that were randomized to placebo, a probiotic, or a symbiotic, which is prebiotic plus bacteria. They gave this intervention for only 15 days, but I guess over 15 days they did see a significant difference in weight loss over 15 days. I don't know if that is clinically relevant at all, but they didn't find any changes in the inflammatory markers seen in the patient's blood. Another larger study, though, found that they gave probiotics over 16 weeks to gastric bypass patients. And in this study, at least, they found that the addition of probiotics resulted in much greater weight loss compared to those that who received placebo. And that in this study, they actually found that there were overall drops in or changes in the inflammatory markers present in these patients as well. But they didn't have any significant effects on glycemic or lipid profiles. So this tells us that there is some hope in being able to influence obesity and the amount of weight loss achieved by bariatric surgery by probiotic and prebiotic interventions. And one additional study also randomized patients to probiotic or placebo and followed them for 6 months, and at least found at the 6-week and 3-month mark there was significantly greater weight loss in the probiotic group, though this was not maintained at 6 months. I think even more interesting than just weight loss is the effects on the microbiome in other settings. And we know that, and this study found that by giving probiotics, patients over the long run actually had decreased what's called small intestinal bacterial overgrowth, which is a misbalance of bacteria in the small intestines. And this has been a research interest of mine. We know that patients with obesity actually have an increased incidence of small intestinal bacterial overgrowth. The symptoms are things like pain, bloating, diarrhea, and flatulence. And this is characterized by excessive bacteria in the small intestines and an increase in the colonic type flora in the small intestines compared to those without SIBO. And patients with obesity naturally have a higher risk for SIBO. And some small case series after bariatric surgery also tells us that the incidence of bacterial overgrowth is much more common in the bariatric population. And so we're doing a small study with Trevor and Alexis, who are really helping us take a deeper delve into our own data. They're helping perform a retrospective study of all patients who underwent bariatric surgery at UC Davis. And they've delved into the records of nearly 100 patients who were tested for SIBO with hydrogen breath testing. They've gathered data on about 60 patients so far and found that symptoms are very common in terms of diarrhea, pain, and bloating, and that so far the incidence of SIBO in our population is at least 25% for patients who have symptoms. So they're continuing to work on data collection and looking at other factors that might contribute to this bacterial imbalance in our patient population. So taken all together, I think this diagram really helps me at least organize my thoughts and next steps that can be done. I think there's been plenty of studies that looked at taking samples from patients who are healthy versus those that are obese or who had bariatric surgery, and being able to do large-scale multi-omics or metabolomic studies to really help us figure out what pathways are involved in obesity and bariatric surgery recovery. And then we can study them in animal models and other ways, and then work on finding interventions that work to help improve patient outcomes. And then there's other ways, other methodologies to also answer these questions by going in reverse, by starting dietary interventions and working backwards into the animal models. And I think this is sort of the next wave of research in our division. Dr. Ali has been spearheading a study currently where women undergoing gastric bypass who have metabolic syndrome or not are getting analyzed in terms of their fecal microbiome and looking at the actual metabolome of these gut flora by looking at blood, feces, and urine and liver samples collected pre and post-operatively to really tell us are there metabolomic changes and signatures seen in bariatric surgery, and how much is that associated with metabolic syndrome as well. And there's really quite a few future projects that we're starting to think about and work forward, including differences in male and female outcomes after bariatric surgery, whether non-alcoholic fatty liver disease associated with different microbiome features, and other projects as well. And we're really hoping to move these types of projects forward with the establishment of the Center for Elementary and Metabolic Science, spearheaded by this awesome team. So I really appreciate your time today and happy to take any questions.
Wow, Dr. Leo, spectacular. This whole topic has bugged me forever of trying to understand this issue, and you've really put it in a great framework. I'd like to call on the division chief, Dr. Muhammad Ali, to lead the discussion and comment on the presentation and the future direction, particularly at UC Davis and research in this regard.
So, Dr. Leo, congratulations on taking a very complicated topic and making it within a 45-minute frame, something that we look at. And, personally speaking, in terms of bariatric surgery, I think bariatric surgery is a very blunt instrument that does not look at patient-specific information. In cancer, it would just be only surgery with no neoadjuvant or adjuvant therapies. And I think you've raised our thoughts today about some of the things that could occur. Looks like you've got some questions in the chat, but I'd like to ask you to start off with one thing. So it's compelling to say that the changes, especially after bariatric surgery, are due to changes in diet, which we know can induce changes in the microbiome. But what's interesting about it is that with the same dietary interventions, without bariatric surgery, you don't get the changes that you experience after bariatric surgery. And so within that, there's a question about the anatomic rearrangement and why that changes. So I wonder if you could, I realize no one knows the answer to that question, but could you speculate on why you think that's the case? And I think you've got some folks on the line that have questions.
Well, just as you mentioned, there actually have been some studies that have compared patients who were on a very low-calorie diet versus those that had bariatric surgery, and the microbiome profiles are actually different between those groups. And so that is already telling us that there's something different about the surgery population. One of the proposed mechanisms is that by rerouting the small intestines and allowing the biliopancreatic juices to flow to the small intestine more distally, and how that affects essentially the motility and transit of the stool and bacteria, but that this also can then influence sort of the optimal growth of different types of bacteria because of the changes in the motility and just changes in sort of the bile pancreatic juice milieu in the intestine. But beyond that, I'm not. There's things that we need help explaining, and that includes the bile acid metabolism and other things.
I think you have some folks on the lines that Dr. Kant.
Can I make a comment?
Sure, Bob.
Hey, sorry. Or go ahead, Mohammed. It's hard on.
No. It's your floor. It's your floor.
Dr. Leo, fantastic talk. And this really is a hot area, as no pun intended. We haven't met. I'm faculty in surgical oncology. We study cancer immunology and immunotherapy. I think it's important to point out how, as you mentioned multiple times, how complicated these studies are. You're dealing with multiple variables and multiple interactions on multiple levels. And we've seen in mice, for example, if you just look at the microbiome between the IRC and Research 3, which is maybe a half a mile apart, you have major changes in the microbiome. So it's influenced by strain, sex, hormonal status, diet, etc. So the studies are very complicated to sort of dissect of what's causing what. And even in the mouse studies, they're giving them antibiotics, which are in the drinking water, which affects how they eat, affects mitochondrial metabolism, affects other physiology, etc. And in the human studies, it's hard to really be completely controlled in terms of your interventions. You're not doing a gastric bypass on patients with normal weight, so you don't know how the gastric bypass is affecting the microbiome in patients who are not obese. So it's very easy to have type 1 error, which can, assess, what's correlation instead of causation. So I just think that's important to keep in mind, even though it's clear that the microbiome has major impacts on, immune physiology, metabolism, all the things that you mentioned. So that's just what I wanted to comment.
Thank you.
So there's a couple of comments and questions. The first comment is from Dr. Brown. He says, one of the challenges in investigating microbiome is that similar to Dr. Cantor's comment on CMV with microbiome, you are investigating a coevolution and have to integrate host genetics and expression, including proteomics and metabolics with environment, including diet, location in the gut, and the actual environment, together with what you see with the actual flora. It's a big chunk of data and complexity to integrate, but the approaches that look at how the microbiome shifts in response to some specific stimulus, such as bypass area, are a great start as they have more limited variables. And then Dr. Adams says, Victoria, such a great overview of the field. Thank you. Can you speak to changes in acid psychology, microbiomes and bariatric surgery?
Can I comment on? So we know that there's been some studies that looked at bile acids and how they change after bariatric surgery, and there are significant differences in both the primary and secondary bile acids that exist in the stool after bariatric surgery. That's probably a whole nother talk on it in its own, and actually an area of expertise of one of our researchers, Bethany Cummings. And so I'll have to defer more discussions to that to her.
Victoria, that's a great talk. And a few things. One, I'm not surprised you're doing such a great job after having known you as a trainee. I like to say as one of the chubby guys in the club that we're all looking forward to fixing this with some sort of stool transplant or yogurt. Yeah, something that you can fix, because us chubby guys need all the help we can get. And finally, I have actually a real question. So what happens when you take antibiotics? Like, what happens to your microbiome? And what happens when, like, we give these preoperative antibiotics now that they're back in vogue for a bowel prep? And do kids that get a lot of antibiotics for ear infections get fat? Because I had a lot of ear surgery and ear infections. I'm blaming that now. Yeah. Because it's not my fault for eating cheeseburgers. It's the antibiotics.
Right. Well, I think antibiotics definitely change the microbiome. I didn't delve into the studies of like single dose versus like multiple chronic doses, but there has been multiple studies that show that it really does influence the microbiome, and we see that, for example, with small intestinal bacterial overgrowth. Patients who are on multiple courses of antibiotics have a change in their small intestinal flora as well as their colonic flora. So I definitely think that those exist, but how much that influences, certain outcomes, it's hard to say 100%. I do know I'm giving my toddler yogurt every day. Whether that, does anything to his future life, I hope so, but that's my takeaway.
Oh, Victoria, one more sort of conversational piece. When you had the diagram there obviously shows that there are metabolites that you can detect in the stool, right? That's that these short chain fatty acids and so forth exist in the local environment. Can you speak a bit to what we now understand to be the xenometabolome, the things that perhaps, but for, talking like my lawyer wife now, but for the gut microbiota would not be in our peripheral circulation, in our blood, because that's a field of interest.
Right. So for everyone else, the metabolome are sort of all of the chemicals and proteins and kind of molecules that are created by our own body, and the xenometabolome are those that are specifically created by the gut microbiome or by the xeno. And so there's essentially been studies that have looked at these different profiles after bariatric surgery or just in general, and we can look at profiling based on the metabolomics done even here on campus, and those signatures really tell us the difference of where those metabolites originate, so from bacterial or from self. And I think this is just a hot new area that sort of needs further investigation. But that's something that I actually have to learn more about in terms of the details.
Dr. Utter asks, the treatment of C. difficile suggests that probiotics do not have the same effect as fecal transplant accepting the fact that feces are extremely complex and inconsistent from a microbiological perspective, have there been studies looking at the impact of fecal transplant before or after gastric bypass, etc.?
That hasn't been done yet, but I think they're still starting to understand, like, well, what is the healthy and ideal microbiome first before they even attempt those types of studies. I think people have definitely done that in mice, but not yet in humans.
Are we at time, or did you? Well, I think just to get everyone done on time, Dr. Leo, thank you very much for a very provocative talk. Thank you all for the questions. And as Dr. Leo pointed out, there's a tremendous amount of outreach and collaboration. So if you have ideas about how the microbiome or metabolome affects any particular field, I think we have now the group of people in place to be able to think about those questions in a more intelligent way. So thank you, Victoria.
Thank you.